Okay, so glycolysis produces 2 ATP, Krebs 2 ATP, electron transport 32 to 34 ATP. Now you might say, why on earth is it producing different levels of ATP? The thing is, all cells actually produce, or different types of cells actually produce different amounts of ATP, okay? And in your textbook you'll see it's even revised down to being lower than that. But the VCAR accepted answer in previous years has been 36 to 38 ATP as the net production. The process is slow, 36 to 38 ATP, CO2 and water is produced, but it is very efficient and the production is almost indefinite. So if we have a look here, what we can see is the electron transport chain produces the most ATP, and we've got these high energy electrons and hydrogens that are carried by our coenzymes, NAD as unloaded and NADH as loaded, which we're going to come back to. So stage 1 of glycolysis is exactly the same as anaerobic respiration, except the pyruvate isn't broken down. So we start off with our glucose, our outputs down the bottom are pyruvate, okay, and then coming out of this stage, going on to electron transport is our NADH. So if we're making ATP and NADH, we also need our unloaded carriers, ADP, and inorganic phosphate, and NAD+. ADP and inorganic phosphates producing 2 ATP here, and NAD+ produces NADH. Doesn't have a P, we're not talking about photosynthesis, so it's just a slightly different molecule. Okay, so NAD, and this is why we need to be clear about this, NAD is the one for cellular respiration. So for our process, glucose is split into pyruvate. We need 2 times 2 pyruvate molecules, H plus ions and electrons, 2 ATP produced. Okay, so for the people that are super interested in the chemistry, glucose is C6H12O6, pyruvate, which we've got 2 of, is C3H5O3. So 2 of those hydrogens from up here have been lost to our NADH.
Do we need to know that pyruvate?
Don't need to know, just know what pyruvate is. Now pyruvate without oxygen becomes lactic acid or ethanol and carbon dioxide, but with oxygen it's able to move on to the next stage. So into the Krebs cycle goes our pyruvate. And our pyruvate has still got plenty of hydrogens that we need to get from it. Okay, so via a number of steps, pyruvate is further broken down to carbon dioxide to remove those hydrogen ions and electrons there. Okay, so the carrier molecules, FAD and NAD, carry electrons and H plus ions into the crystallate because that is where they help produce lots of ATP. Now you don't need to know this, but our NADH produces 3 ATP for whatever reason. It will be to do with the proteins involved in the membrane, and our FADH2 produces 2 ATP. You don't need to know that, but that's why we're getting a range of ATP produced. It also might produce less ATP depending on where it actually enters that biochemical process, which we'll come to. Okay, so NAD with our H plus and our electrons, okay, produces the carrier molecule, and then we've got our NAD plus. I would just show it like this. In we're getting 2 electrons and a hydrogen ion forming NADH. So it's just like the truck now, it's picked up whatever it's transporting and then it drops it off at the next stage. So these are our really basic definitions, okay? Processes. Breaks down pyruvate, releasing some carbon dioxide molecules, creating some ATP and some carrier molecules. Electron transport, we're going to talk about shortly, that it uses the H plus ions and electrons to make the ATP and uses oxygen to create water as a waste product. So coming out of here as well, we've got our NADH and our FADH2. All of these molecules, they're actually, I had to do research, they contain carbon, hydrogen, oxygen, nitrogen, and phosphorus. So therefore, what biomacromolecule would they be? Carbon, hydrogen, oxygen, phosphorus, and nitrogen. Shopping. They're a nucleic acid. So those carrier molecules, okay, or the coenzymes, whatever you want to call them, they're just a nucleic acid. You don't need to know their chemical composition. You just need to know that they carry hydrogens and electrons.
Does FADH also come out of glycolysis?
No, it doesn't come out of glycolysis, only out of the Krebs cycle.
And is it going into electron transport?
It goes into electron transport to drop off the hydrogens and electrons, which is the bit that's going to help produce our energy. So now we've ripped all the hydrogens and electrons off. That's what's going to produce our energy. So into this stage are our NADH molecules and our FADH2 from glycolysis and Krebs cycle. Okay, and it drops off the hydrogens and electrons. So coming out of here, into here is also the oxygen. Coming out of here is our water and our unloaded carriers, NAD plus and FAD plus, which will go back to glycolysis, back to cytosol and mitochondria. Oh, cool. Good idea. You can use that one. Alright, so we'll go through stage 1 and 2 again, and in electron transport we're going to go in a little bit slower because that's the one that provides the most confusion. Yes. What do you mean? Yeah. Well, because they have the enzymes that allow that to occur. Electron transport has to go across the membrane and it has to create a concentration gradient. So that's why that one has to happen at the cruise stage. The matrix is a nice fluid, so that's just those other reactions are happening in there because it's within the mitochondria where the enzymes are that help Krebs cycle happen, but it's a fluid, so it's good for lots of reactions to it. Yeah, pretty much. The photosynthesis though, the grinder is because the chlorophyll's down there. Yeah. Sabrina?
I'm just confused as to what the FADH is.
FADH is another carrier molecule. So just like NAD plus and NADH, okay, FADH2 is doing the same thing. So it is also just a coenzyme with the role of actually carrying electrons and hydrogen. It's not specifically mentioned in the study design. However, it is part of cellular respiration. So its job is would be another molecule, slightly different, that in Krebs cycle would also collect up some of those hydrogens. Nathan, do you have a question?
I was going to ask you the oxygen function, that's just like how that and maybe different.
Yeah, so the oxygen will come in, diffuse into the cell from high to low concentration. Alright, so glycolysis, we've got the glucose leaves 2 making our 2 pyruvate molecules, and it produces some NADH as well. Okay, and 2 ATP are produced and it happens in the cytosol of the cell. Does that make any sense? Yeah. Okay, here we have a diagrammatic picture.
There was one of these on
The homework.
yeah.
Yeah.
So, yeah.
What's up though? The
Is that
P's. Here's our glucose. I would say that they're phosphates, inorganic phosphates that probably help with the production of ATP, but they might just be used for other processes. You don't need to know it, but it's just to get the idea that it's a bit more complex than we realise. So when we actually start this reaction off, it uses some energy, but then more ATP is produced, so the net production of ATP is 2, but technically it uses 2 and produces 4. Okay. Once we've made it's been split, we then steal some hydrogens off it and we make our pyruvate molecules here. Okay. You do not need to know it, but it's just to show that it's not as simple as chopping the glucose in half. What happens after glycolysis? It depends on whether or not oxygen is present. When oxygen is present, pyruvate can go on to the Krebs cycle to produce some more ATP. Okay? In the mitochondria is where this occurs. Without oxygen, we have fermentation occurring or anaerobic respiration, which produces alcohol and CO2 in yeast and plants, and lactate or lactic acid in animals. Yeah, I've only ever seen it referred to in a VCAR sense, yeast and plants. Alright, so Krebs cycle. In comes the pyruvate. We agree with that. Out comes some ATP, some carbon dioxide, and some carrier molecules. It occurs at the matrix. Pyruvate from glycolysis enters via diffusion from high to low concentration. Krebs cycle completes the breakdown of sugar by removing carbon dioxide, so all the H+ ions and electrons are released to be used in that final stage of electron transport. Okay, if I break a bond and remove hydrogen ions and electrons, energy is released. What's going to carry the H+ ions and electrons? Our carrier molecules, NAD and FAD+. We've got 2 ATP, and they can also be referred to as loaded acceptor molecules or loaded carriers. They are loaded now because they have a hydrogen and an electron attached to them. Krebs cycle produces these as our outputs, which we already have. So here again is the Krebs cycle, much more complex, okay, than we think. Okay, the acetic acid is the step in between pyruvate and entering the Krebs cycle. So if you ever see acetic acid or acetyl-CoA, they're just talking about the intermediate step involved there. You don't need to know pyruvate as an input is fine. It goes around and around and around and around and around until we produce some carbon dioxide. okay? And then we produce some ATP. The amount of ATP occurring and produced though is 2 ATP. I think that's just the order of the steps that we're going on there. Again, don't need to know how that works, okay? Just need to know inputs and outputs. And even simply saying loaded carriers are produced and not knowing the difference between the FAD and the NAD, FAD and NAD is fine. So now this is the bit that's confusing. Here, all the loaded carriers drop off the hydrogens and electrons. And we're going to watch a little video to help us understand the process of what happens. But what I want you to realise is, what happens if we've got a membrane? I'm just going to draw this here and then delete it. What's going to happen with a membrane if I've got all of the hydrogen ions here and I open up a hydrogen channel? Where are the hydrogens going to move? All the way down from high to low concentration. So we agree with that? From junior secondary, if we've got a negatively charged particle and a positively charged particle, what's going to happen? They're going to be attracted to one another. And so the negative electrons help form a concentration gradient, which is going to help form our ATP. So in our little movie, that is what we're going to see. But again, remember, it's beyond the course. So I'm going to remind you at the end what is important for you to know.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain:
I'll explain what you need to know in a
dehydrogenase,
second, but you definitely don't need to know all
cytochrome
of this.
BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, 2 mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. 1 hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation.
What? Any sense?
This animation illustrates 2 full cycles.
Very straightforward. We're just going to go back. I'm going to turn the sound off. We're going to talk about it again. So they talked about all the components, and then they started off here. We've basically got a membrane We've got a membrane, and embedded in the membrane are transmembrane proteins, okay? They are just proteins that are involved in this process. Some of them are cytochromes, some of them are other enzymes, and so on. Coming along here is our NADH. Our NADH has the 2 electrons and 1 hydrogen. So they carry the hydrogens, and when the electrons are released, the hydrogen is released as well. But when the electron hits this protein, that protein then becomes negatively charged. And when it becomes negatively charged, the positively charged hydrogen is going to be attracted to it, and it's going to allow the hydrogen to transport across the membrane. Okay, so we're just basically saying electrons are being passed through the protein, transporting the hydrogens to the other side of the membrane. Those electrons move on, and the same thing happens here. So again, negatively charged, pumping hydrogens that are positively charged to the other side of the membrane. The final thing that happens is where the water is produced. So once these hydrogens are used up and they fall across the membrane in that image later on the right-hand side, there's lots of waste products down here, some waste product hydrogens. So what happens is the oxygen molecules, okay, the oxygen molecules combine with the electrons and some hydrogens to make water, and more hydrogens are pumped across the membrane. So you just need to know oxygens combine with the hydrogens to make water. Down the bottom, here are our water molecules. But now what we can see is a concentration gradient, because we've got lots of hydrogen above the membrane, not much hydrogen below, and the potential energy that's in that gradient, okay, if you think about it like a water wheel, okay, there's lots and lots of water, the water will make the water wheel turn, okay, the hydrogens are going to make the ATP synthase produce our ATP. So we can see that now those hydrogens moving across the membrane again via that enzyme is what's actually generating the energy. And we can see down the bottom, over here, there's our ADP, and inorganic phosphate, and that is what is synthesizing the reaction and storing the ATP. Okay, so if we're keeping it really simple, for our process, electrons are passed via a series of proteins in the membrane. Pumping H+ ions across the membrane. Fall of H+ ions through ATP synthase, and this is even more information than we need. Synthase produces ATP. H+ electrons and oxygen combine to form water. Again, is really beyond what the study design is telling us we need to know. But it helps us understand the complexity of the process, how complex that process is. So when they move from high to low concentration, so you could say fall, or you could say a hydrogen gradient is created, and the hydrogen's moving through the ATP synthase produces the energy. Some of us might not be feeling very comfortable with all of that information. So another way that you can present the same information on the board could be like this. Okay? Much more straightforward. Does everything that the VCAR recommends. So I've put it up on the Canvas page. But if we have a look, it's just got the things we need to know. The location, the reactants or inputs, the products or outputs, the carriers, the ATP, and then it shows our carriers here as loaded or unloaded carriers. Overall goal, get the hydrogen and electrons from the original glucose, okay, because that's what generates the ATP in the electron transport chain.
So is the hydrogen gradient make the ATP synthase go faster?
Well, it basically is what's almost required. The gradient provides the energy, I suppose you'd almost say, or the gradient is the potential energy when it's moving from high to low concentration that's allowing the energy to be stored in ATP. So I'd say potential energy, hydrogen gradient, okay? So the hydrogen concentration gradient difference is what is allowing ATP synthase to function, producing our ATP.
So like the difference in potential energy.
Yeah, well basically the concentration gradient creates potential energy. So if you go back to your little video, yeah.
So the potential energy helps the ATP synthase.
Function to produce the ATP. Yeah, so if we have a
These
listen
4
again.
electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP.
Okay, so hydrogen ion concentration gradient, the potential energy in the concentration difference is what allows ATP synthase to make the ATP. But for us, inputs and outputs are the key things that we need to know. Questions?
So it's like kind of like a seesaw.
It's kind of like a seesaw. Yep.
Yeah.
So it's basically going negatively charged. When it's negatively charged, hydrogens can get on the seesaw and then they're pushed up to the top. Then once they're up the top, they have more energy than they did at the bottom of the seesaw. So then them crossing the membrane at the end is what generates the energy.
Yeah. So that makes all the ATP.
That's what makes the ATP.
Yeah.
The fall of or the movement of the hydrogens from high to low concentration. Seesaw, okay? Seesaw becomes negatively charged with the electrons, moves the hydrogens up. The hydrogens now have more energy than they did before, okay? And that concentration gradient difference produces the energy. Questions? In terms of the stack, you're only going to be assessed on what the study design states, which is the inputs and outputs locations of aerobic respiration. But it's just going into that little bit more detail there.
Yeah, that's why I said
SE.
Yeah, that's why. Yeah, just say it right.
SE.
I looked up.
Alright, so electron transport. This drawing shows the mitochondrion cut lengthwise to reveal its internal components. The intermembrane space, the cristae, which is the membrane itself, and the matrix. Here is exactly the same thing, but we've got our electron carrier here. So the electrons are passed via these series of proteins, and because they're negatively charged, they're pulling the hydrogens up across the membrane, okay, creating that potential energy with the concentration gradient. The hydrogens moving through ATP synthase is what is used to produce the ATP. Okay, so you could say something like this. The accepted molecules enter an electron transport system with the inner mitochondrial membrane where electrons are transferred through a series of cytochromes until they are finally accepted by oxygen. When the electrons are accepted by oxygen, the oxygen becomes slightly negatively charged, which means it's going to attract the positive hydrogen and form water. Okay, so the oxygen is almost like the mop. Okay, it's going to mop up and form all of the water. Electron transport releases energy to make ATP. The molecules of electron transport are built into the inner membrane of the mitochondria. The chain functions as a chemical machine that uses the energy released by the fall of electrons to pump hydrogen ions. So it's really, there's energy in this process here, this fall of electrons, which is going to be used to push all the hydrogen. So key things to take away with aerobic cellular respiration are our inputs, outputs, locations, and ATP produced. Has everyone got what they want on that PowerPoint slide? Yep. Okay, so here's another way to do it in a little table like so, okay? A really good way that you can then just quickly do that, okay? So glycolysis creates electron transport. The other way that you can be reviewing it, and then we're going to look at some questions, is like so. So if anyone wants to take a picture of that, you can, but it is also up on the Canvas page as well. Okay?
Do you need to know how much of the carrier molecules?
You don't need to know the amount of carrier molecules produced, but I think it's 2 NADH, 2 NADH in the first, 8 in the second, and 2 FNN. So if you want the values,
Wait, so there's two,
well, you can, but you don't need to know them, and there's 2 of the molecules. Alright, so let's look at some sample questions now. Let's look at some sample questions now. Mitochondria, question 15. A, contains circular DNA. B, are the site of glycolysis. C, prepare proteins for the Golgi complex. D, produce 2 ATP molecules of glucose. A is the correct answer. So circular DNA, okay? Circular DNA, and that is evidence of their bacterial origins. Yes?
Yeah.
Ruby, that was a good one. This one, I'll read it out to you. A student was asked to identify differences between the overall processes of photosynthesis and aerobic respiration. The student prepared the table below to outline the differences. The only correct comparison listed by the student is A, photosynthesis, exergonic, aerobic respiration, endergonic. No, why not? Photosynthesis is endergonic and aerobic respiration is exergonic. So that is incorrect. B, all stages occur within chloroplast. Well, that's right. But for aerobic, all stages occur within mitochondria. No. C, electron transport not involved.
Oh, no, but it's just not.
It's just not a stage. Why is it involved? Because NADPH transports electrons. So it's still electron transport. There's just not an electron transport stage. And aerobic electron transport is involved. So that one is still incorrect. So D should be the correct answer, but let's see if it is correct. D uses water as a reactant in the first stage. Yes, because water is split by sunlight. And aerobic forms water as a product in the final stage. So a little something that people have gotten tripped up by is they've chosen C. But hopefully they got to D and then said, oh, hang on, I know D is correct. So there's got to be something I've got to come back to at least check this question. Alright, talk with the person next to you about this one. Pretty straightforward. What is the energy yield per molecule of glucose as a result of aerobic respiration? 36 to 38 ATP. And then the next bit, we just have to use the diagram and then go on to the questions on the next page. So the diagram's got glucose going in, glycolysis, Krebs cycle, electron transport, and something going out. What's coming out of the electron transport stage? Water or 32 to 34 ATP. So within the cell, where does electron transport stage happen? In the cristae of the mitochondria. So don't just say mitochondria. Be specific. Then describe what happens during electron transport stage. In your answer, include the name of product said. So you could say something simple such as hydrogens and electrons are involved across the membrane to produce ATP. Water is the product set. Okay, so look at what they've said. Description. Electrons are passed along electron acceptors. Oxygen captures electrons which are combined with hydrogen. Carrier molecules gives up hydrogens as it's passed along. That's a really explicit description. Almost too much information. It's 1 mark that they're giving. So even if you said something like oxygen combines with electrons and hydrogens to produce water and ATP is produced, that would be enough to gain the mark. Yes? Yes. So when you're writing the equation, you do need to show the ATP produced. So I would be doing this. C6H12O6 plus 6O2 forming 6CO2 plus 6H2O. And then I have here plus 36 to 38 ATP. No, you can have it coming out of the arrow if you want, but if you've got it over that side, you need 36 to 38 ADP as well. Before you go, I know that some people are very busy on their holidays, so your holiday homework, I will give you this information via email, but for people that are wanting to get ahead with their holiday homework, it's also going to be good provision for your SAC, is the 2 area of study topic exams, okay? Area of study 1 topic exam 1, topic exam 2. Both need to be completed for your holiday homework. The other thing I want you to be doing on your holiday homework, is to be working on these posters. So we have now covered, in our lovely little green book, we've now covered 1, 2, 3, 4, 5. So we've only got 6. We've only got 3 to go. Okay? So on the holidays, I want you to really start putting your posters together for each of the study design dot points. Okay? So that's your holiday homework. Homework for tonight. You've got some Ed Rollo and some practice stuff for your snack. Can I just show you one more thing? One more thing is here now on the cover page for Unit 3, you've got your SAC 1B revision. You've got the revision sheet you can click on, and you've got the key knowledge and the skills and the terminology, and you've got some practice questions. So that is your homework to make a start on that. You might want to save your practice questions for the weekend. There's also your other photosynthesis questions that you've done. I'll try and confirm with the other teachers if we've got any other practice questions to do. Yes, logbooks need to go back, but just hand them to me as I come past and I'll put them away for you, and then you can fill. Where's your eyes? Just come up the front and then I'll just take you and stuff.
Bye. Enjoy going home.
Okay.
I couldn't keep reading up to the bloody bullet journal.
See you later.
Bye.
I'm so bad.
Brandon, you okay? Yes. Are you? Yeah.
And you got it next time?
Whoa, I can't. What? Hi. Oh, I'm gonna have to fold out my blazer. What's the best way to fold it without any crinkles? There's no way to fold it without getting any crinkles. There's like no room in my bag for any
This
of
hurts my, and you can't really iron a blazer, can you?
Yes, you can.
Can you?
I guess, I know, I never wear my blazers.
Oh, I'm gonna iron.
Okay, I can't be bothered. I kind of stuff this in my bag because I don't want to stuff it up. Look what I bought today.
Hi. Put that on.
See what Denver got.
Yeah, you can come in.
Hi, Miss Paul.
Yes. Hey, hello. Bye. Yeah. How was it? Good. I only found 2 obstacles. What were they? The fortress, which was the climbing the wall, and then the overhead, and then the other part of the wall. I got onto the overhead bit, but I just didn't have anything left to get around. And then the spear throw is what I was doing. So I threw it, but it didn't stick in.
Hello, ladies. How are you?
The class is just unwell, so I'm trying to because it's just an extended. Got your lollipop.
Like only when you say
I wouldn't,
Another UM question and another article which we're going to put in. So we got exam question and then article 3. Emily gets the lollipop. Wrong one. My brain. Okay.
I've been painless this whole day.
So Chloe, yesterday, I couldn't be.
I'll let her. So, you get to go again. And PH. And ATP produced. I was pretty, yes, you can eat your lollipops. If you didn't get a lollipop, you get a second chance today. Can you make sure you fill it in without referring to the corrected sheet, and then go on to the exam question, and then go on to the article? So, boys, Annie, sorry, boys. Oh, what am I doing? I'm going to say lollipop? Yes, yeah. But you get a second go. Kelly's got Gabby, what's going on? You missed out on one. So can you do your second one for your lollipop today? Ellie, lollipop.
I'm sorry, I'm like, thank you.
Oh no, minus one. That's okay. Yeah, this is yours.
Probably mine.
Oh, wait.
Can you do that for me now? Yes. Oh, right. I was like, hang on, I've got it. But yeah, no, right. You might get a lollipop later. Are you serious?
Oh.
Does everyone have a copy of the RAT article? Yes. Yeah? True. Does Lucy's spot have one? Yeah. Yeah, so do you want to get now?
Sorry, let's tell you this one.
Okay, I'm bringing around your sacks as they have been marked the second time. If you picked up the marks for question 4, it will be in green pen. So, some people already had the 2 marks, like, yeah. So, if you've got any questions about that, please come and see me. So some people's I'm just giving back, but some people did pick up some marks. even though remember on the exam, you won't get the marks, but on the SAC, you've got them so that we're consistent and fair with everyone. Yeah. I will check. Thank Because I wouldn't give the marks for the experimenter error, right? So that's how I marked it. So then Mr. Torstenson said to his class, Miss Paul didn't give the marks for the experimenter error, rah, 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 rah. But you, student X, you got 2 marks. What marks did you get? She reads out her answer and she reads out word for word what I wasn't giving marks for. But for some reason, I gave marks for that question. So I had a brain fade just for that one particular question, for that one particular student. And so as a result, Mr. Torstenson said, everyone has to get, said that has to get the mark. So I just collected everybody's back up and remarked everybody. So you would not get the mark for saying an experimenter error on the exam, but on the SAC you would get the mark provided that you made the link to the effect on catalase activity or substrate concentration or blah, 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 blah. So that's why it was completely just a human error, which happens, hence why your exams at the end of the year are marked. Correct. So no one's marks should be removed. That's what I said. I was like, why can't we just take the marks off that one student and then it's all sorted? But this is the fairest way. So the other way we were going to do was to omit marks and so on, but this was the way that we agreed was the fairest. So. And the main thing, guys, is that we're consistent and fair across all of the classes. And I want you to have a look at this. There's something that read the names of these stages carefully.
Have you been marking these today?
Which one? Yeah. Lara, need your broken lollipop because I still got things missing. This bit. And you can take back to it, so you get a broken one. So once you are done with the exam question, it is double-sided. Can you please come and grab your logbook, read the article, and then put some little summary notes in your logbook about that article, and then we will go on from there. Yeah. So you gotta make sure in your description, I am being a bit of a mean, but detail. Oh, I gotta be so mean. Yeah, I'm jealous. I like yellow. You left a letter out. N-A-D-P-H, not N-A-P-H.
Oh, surely not.
Oh man, how is this possible? Leave my desk for 5 minutes, 8 emails. No, seriously, I got interrupted 4 times at the same time yesterday. So I was with a student, and then a teacher interrupted me, then a teacher interrupted that teacher, and then a teacher interrupted the other teacher. There were 4 people, and I was just like, yeah. Basics. So everything you can pretty much do, provided that you get ethics approved. So like, correct. So they're going to have an ethics committee at the university that they'll have approval for. So one of the things that I came across at uni that I thought was exceptionally amazing that was accepted were Tamar wallabies. They looked at milk production, but milk production is effective for wallabies are euthanized, so the only way that they can kill them is by a blow to the head, because then it's not causing any impact on the hormones that they need for the milk, whatever the projects that they did. And so as a result, like in third year uni, for example, we had ethics approval as uni students to look at hormones produced by the pituitary, and you would actually give a radioactive marker that would bind to their pituitary gland. Then you kill them, basically cut open, pull off their skull, pull their brain off, and sitting over the brains of the pituitary, so then you can harvest the pituitary and prove that that's where those receptors were located, because the radioactive marker is found at that location. So there's a lot of things that you can pretty much do, provided that you get the ethics approval to do it. So for example, one of my friends looks at spinal cord injuries, and so he intentionally inflicts spinal cord injuries on mice and rats, and then kills them, and then takes tiny slivers of their spinal cord around the injury site to then see the effect of stem cell treatments and bits and pieces on those cells. So it's pretty horrifying when you learn about it, to be honest. Depends what subject. If you were doing invertebrate structure and function, then I would say E. But if you don't choose that as a subject, you probably Alright, let us go through and correct that exam question if you haven't done it yet. We'll talk about it, then we'll talk about the article, and then we're going to jump into aerobic cellular respiration, which should be pretty straightforward for you. Up on the Canvas page there is your SAS 1B revision now, which has some practice SAS style questions for photosynthesis and cellular respiration, and all the bits and pieces you need to review for that. So please keep your eye out on Canvas for that. Not mine. Alright, so question 7. The diagram below shows the chloroplast and X is labelled. Describe the chemical changes that occur at location X and light is present. So what is X? Grana composed of thylakoid. Okay, and at location X, what chemical changes occur? Yep, water is split, okay, into oxygen. To get your mark, water is split into hydrogen ions, or NADPH is formed to get you a mark. And what else is produced when we split water? ATP is produced. So you can have a mark for oxygen, a mark for hydrogen ion, or a mark for ATP. So any two out of those three will get you the mark. Electrons as well, yes. The graph below shows the rate of carbon dioxide exchange between the leaf and its external environment as light intensity is altered. All other variables are kept constant. So we can see that the reaction rate is occurring. So we've got this -5 CO2 used. So how can we have a negative amount of carbon dioxide? From cellular respiration. So it's that idea, okay, that at 0, which they call the compensation point, don't need to know it as being called that, but at 0, the rate of photosynthesis is equal to the rate of cellular respiration. So at point M, the carbon dioxide produced by cellular respiration is being used by photosynthesis. Beyond that point M, more photosynthesis is happening than cellular respiration, so the cell needs to take in carbon dioxide from the atmosphere. What are some of the reasons why the graph would become horizontal from about 600 absorbed light units? Do we think? What?
Carbon dioxide becomes a limiting factor.
Good. Carbon dioxide becomes a limiting factor. What else? What's another thing that could be a limiting factor? The amount of light, well, absorbed. So the light absorbed, you can see down the bottom, the absorbed light is technically still increasing, but light absorbed by the chlorophyll, or the chlorophyll can't absorb any more light. Yep. So that would probably link into your chloroplast, but it wasn't an answer VCAR had.
Like the temperature the plant's in.
If the temperature was a limiting factor, you'd see it denaturing and going, oh, I can see what you're saying, but no, I don't think they had temperature as a response. So lack of water, lack of CO2, lack of enzyme, or inability of chlorophyll to absorb any more light. Were the ones that they accepted. But yes, if temperature was a limiting factor, it would start to, well, the CO2 use would decrease because the enzymes would stop using that. If CO2 use is a rate of reaction, a measure of rate of reaction. Does that make sense? Is temperature clear?
Yeah.
Okay, so Rubisco is an enzyme found in chloroplasts. Its normal function is to catalyze the reaction in which carbon dioxide is a substrate. Which reaction is carbon dioxide a substrate? Light independent. In certain plants where the level of carbon dioxide is low, Rubisco uses oxygen as the substrate and releases hydrogen peroxide and ammonia. What do you think the effect of hydrogen peroxide and ammonia is going to have on a living tissue? Be harmful, toxic, not be very good. Okay, so explain why it's beneficial for a plant to have a high level of carbon dioxide for 2 marks. What is increasing carbon dioxide concentration going to do? Increase the rate of reaction, specifically photosynthesis, producing more. What does the cell need? Specific molecule. Not ATP, so we're not talking about cellular respiration. Glucose, okay? Increased carbon dioxide concentration increases the rate of photosynthesis, increases glucose for the plant. The second reason why carbon dioxide being in high levels is good is so that Rubisco doesn't use oxygen as the substrate, and therefore toxic products, hydrogen peroxide and ammonia, are not released. But you can see 45% of the state got 0 for that task. I think as long as you've got a reasonable benefit for the plant, you probably would be accepted. But I would say the main reasonable benefit for the plant for photosynthesis is glucose. So glucose is going to be a better. One reason would get one mark. It says either of, actually, but I would suggest that you need 2 points. But yeah, I suppose increase the rate of photosynthesis 1 mark and therefore greater production of glucose would be the 2 marks there. And because it says either of, it really states it only needs 1 point. But I would still be probably making 2 points. Alright, so if we jump in and have a look at that article now, the first sentence says, the naked mole rat can cope fine with oxygen levels low enough to kill us. It can even go 18 minutes with no oxygen at all. So, what's interesting about this is if you keep looking, they stopped breathing after a few minutes because there's no oxygen. But their hearts kept beating and they revived as soon as they were returned to normal air. They come back to life without any apparent problem. Yes, so they're different to diving mammals, such as whales, because diving mammals store oxygen in their tissues to help them survive. But naked mole rats, by contrast, live underground in colonies of up to 300 animals where oxygen is likely to be limited. The tunnels that link colonies to the surface are narrow and can get blocked by heavy rain. What's more, the animals tend to huddle in nesting chambers. They like to pile together in a big heap of naked mole rats. That was awful. Has anyone seen a picture of this? It's disgusting. That's all that I'm going to say about it. So how do they cope with the resulting lack of oxygen? Partly by minimising their need for it. Naked mole rats have a low metabolism and burn little energy heating their bodies instead of staying at the same temperature as their burrows. But they also go into a sort of suspended animation in zero oxygen. But the team found a clever metabolic trick that helps the mole rat survive. So this is the really interesting part that links into our study design. Animal cells get their energy from burning glucose. When there is no oxygen, these cells must use far more glucose to get the same amount of energy, and the process produces lactic acid, because that is anaerobic cellular respiration. Lactic acid we know isn't good, and we know that it only is producing 2 ATP per molecule of glucose. High lactic acid levels can kill cells, so a feedback system soon kicks in to shut the process down. But if cells use the sugar fructose instead, they can bypass this system and keep producing energy. And that's exactly what naked mole rats do. They release fructose into their bloodstream when oxygen drops too low. The sugar is taken up by the heart and the brain cells to keep the critical systems running. Okay, so fructose doesn't get broken down to produce the lactic acid, the harmful lactic acid byproduct. But can we see how that links into anaerobic respiration and understanding? Yep. So again, 1 or 2 sentences as a summary note. Put the date up the top, which is the 21st of March, in your logbook. And then once that's done, I'll collect it up at the end. And then we're going to get into aerobic cellular respiration. Which provides enough energy for them to survive for at least, well, for the 18 minutes without oxygen, because they're going to be able to produce the energy without the harm of the Here's the news out of Kenya overnight. The last white rhino, male white rhino, has passed away. Now, well, there's 2 females left, but both females can't carry young. So they're not extinct, but they're basically unable to survive without some sort of intervention from humans. So what intervention that is being proposed, because as I was telling the other class, my life has gotten so boring now that I listen to talkback radio on my way to work. I listen to Triple J and I just don't like the new morning voice, and everyone else at breakfast radio is boring. So 774 it is. So they were interviewing a person, but they were talking about how they're going to try and bring back these animals. They've stored some of that rhino sperm and other male northern rhino sperm. The females, unfortunately, can't carry young for whatever reason. They've had high percentage of miscarriages, but they do have some eggs. So they're starting a crowdfunding campaign to actually, via IVF, take the egg and sperm and fertilise them, but then put the egg and sperm into a southern white rhino, so a different species.